So we're going to focus on 3 areas of this topic of the kidney today. We're going to look at the structure, the adaptations of the nephron and the blood vessels that are associated with it. We're going to look particularly at those 2 big processes which you'll know about, ultrafiltration and selective reabsorption. We're then going to look at the loop of Henle, which its main features, I mean, you will have heard it called the hairpin countercurrent multiplier, so we'll look at that, and then we'll also look at negative feedback as regards osmoregulation. We haven't got time in today's session to sort of do all parts of the kidney, but I've tried my best to sort of cover as many bits as I can for you. Okay, so let's start then by thinking about homeostasis. And I always think about homeostasis as the maintenance of a constant internal environment. That was what I was taught at your age, and that is what I still remember. And homeostasis is this process where actually there are natural variations in levels, in set levels in the body. But what happens through a process called negative feedback is that the fluctuations, so if there's a fluctuation away from an ideal value or a set point, negative feedback brings the level back to the ideal or to the set. Now, there are lots of examples of this in the body. For instance, our core body temperature, you know, whether you're 36.5, 37, whatever it happens to be, you know, the ideal, if your body temperature moves away from the norm, the ideal, the set, negative feedback will bring it back. And that is the same for pH, water potential, all of these sorts of conditions in our internal environment. They will naturally fluctuate, but negative feedback is a process where we put a corrective mechanism in place to bring that value back to the set. Okay, so that's homeostasis. Of course, it's so important, as I've said there at the top, because it's going to allow cells and metabolism, which is all the cells' reactions, to function efficiently for us. So how does this corrective mechanism work then? Well, I've sort of said a little bit, but what we need are receptors, which are going to detect the internal environment to monitor any changes there. And then if there is a change, the receptor will need to communicate with the coordinator. And, you know, we're used to, you know, seeing the coordinator as brain or spinal cord. Then the coordinator will communicate with effectors. They will be muscles, they will be glands perhaps. And then what happens is a response is put in place to bring the level that's gone away from the ideal set value back to that set point. And of course, the corrective response needs to stop there so that we don't have a positive feedback situation. What happens is the corrective mechanism is put into place, and then it'll be back then to the receptor that'll detect that the internal environment has returned to the set. So for instance, a hormone may have been produced, and then because of the corrective response, then that hormone may not be produced anymore, or may be produced in a smaller concentration. So excretion, which is what the kidney does and is part of, is the removal of this metabolic waste. So that we're talking about waste that's been involved in the chemical reactions of the body. So let's be clear about that. And actually, let's think about some of the substances that are excreted. We're going to focus on urea today, and urea is excreted not only in urine, as we know, but also in sweat. But we've got other things as well, like carbon dioxide. And bile pigments are interesting because bile pigments are made as a result of chemical reactions in the liver, but they are then excreted through the bile duct into the digestive system, and then they are moved out to the body in digestion. But please be careful that there is an important distinction between these two. So just quick little recap then. If we think about, I've mentioned osmoregulation at the start, I've mentioned excretion, I've also mentioned a variety of body fluids. And so really, if I was going to sort of match these up, osmoregulation would be control of the water content, the composition of our body fluids. Excretion will be the removal of these waste products, and we're talking about nitrogenous products today, like urea, and body fluids that we'll talk about will be things like blood and tissue, but not to forget also that the lymph, as part of the lymphatic tissue, is also a body fluid. So where does the urea come from? I mean, this needs to be filtered by the kidneys, but it's got to come from somewhere. So what happens is that we cannot use excess amino acids. What happens is that they have to be removed from the body. And in fact, what happens is they're kind of dismantled. So amino acids will reach the liver from the small intestine in a special little vein called the hepatic portal vein. And then what happens is in the liver, the amino acids, the excess amino acids from any excess proteins that a person's taken in their diet, will be split into two parts: organic acids, which can then be fed into part of Krebs as part of respiration, or converted into ammonia. Now, we then need to actually get rid of this ammonia, or sort of certainly convert it into something else, because it is so toxic, and that's what we do. We actually convert it into urea. So that's what's happening in the little box at the top there. You can see that the amino group has been removed, ammonia has been formed, and what we need to do now as part of something called the ornithine cycle is to actually take the ammonia and convert it into urea. Okay, so you don't need all of the details that I'm showing you there as part of the ornithine cycle, but it's just there to show you that the amino groups, once converted into ammonia, are then converted into urea, and it's taken in the blood plasma to the kidneys. I've put this image in there, or series of images in there, just so that you're aware that you need to be able to label, you need to be able to recognise parts of the urinary system, part of the kidney, the outer cortex, the inner medulla, the pelvis there, the ureter, etc., etc. So that's just like a little reminder for you to say, please make certain, if you're given a diagram or there's a sort of a drawing that you may need to recognise, or an electron micrograph as part of maybe some practical papers, that actually you're able to recognise it for yourself. So I'm not going to go into the detail now, but they're there for you. What I'm now going to focus on is actually the main processes that take place in the kidney, and the first one is ultrafiltration. And so this is what happens in the Bowman's capsule. You can see the Bowman's capsule there, and we'll start by talking about the fact the blood flows into the Bowman's capsule in a branch of the renal artery. It's a tiny branch of that artery called an arteriole. Now, that branch that actually arrives at the first part of these tubules inside the kidney, called the nephrons, as I'm sure you'll remember, they are actually called afferent arterioles. And the feature about the afferent arteriole that you need to remember is the diameter is much larger than the diameter of the arteriole that leaves the Bowman's capsule, and that's called the efferent. I always think efferent has had its effect, that's why it's going on its way, but that's afferent and efferent arterioles. So sometimes the Bowman's capsule is called the renal capsule. You may recognise it as that name, but essentially what's happening here is, because of these differences in diameter, a high hydrostatic pressure is developed. And of course, that high hydrostatic pressure then forces molecules that are small enough through the knotwork of capillaries that we know as a glomerulus, through into the Bowman's capsule, and from then into the next part of this nephron called the proximal convoluted tubule. So the sorts of substances which can be forced through, we're talking small molecules like water, glucose, amino acids, salts like sodium and chloride ions, urea, but remember that it's not possible for large molecules like large proteins, or also cells, red blood cells, white blood cells, to be pushed through, because they're way too big to be pushed through. Okay, so what we end up with after ultrafiltration is something called the glomerular filtrate, because the fluid created has actually being produced here as a result of the pushing through from the glomerulus. So just want to sort of go into a little bit of detail because I'm sure if you see that diagram there, you can see that the Bowman's capsule has a layer of squamous epithelium called podocytes. Now, squamous epithelium, I always like to think of like patio slabs. They're sort of fairly thin. They're quite broad, but they're very thin. So what happens is in the Bowman's capsule, it's got this layer of squamous epithelium, and it's also got these finger-like extensions. And those are called pedicels, and what happens is they kind of wrap around the blood capillaries there in the glomerulus, and they kind of sort of pull or make certain that the blood capillary is very, very close to the Bowman's capsule. So the blood capillary itself is also 1 cell thick, so it has an endothelium, and it also has a basement membrane that we like to call a molecular filter. So there's 3 layers there, but the podocyte is sort of bringing all 3 layers close together. So I think I've probably said everything there, apart from the fact that the capillary that you can see there in pink in that diagram there, that indeed has little pores, which we call fenestrations, and they're small, but they're large enough for these substances that we've been talking about, the glucose and the amino acids and the water and the urea, to be able to travel through from the Bowman's capsule through into from the blood capillary, into the Bowman's capsule, and then through into the nephron. Okay, so just thought I'd show you some little images there. There's an electron micrograph, and you can see the pedicels there wrapping around the blood capillaries. So you can see that there are these little lines, little fingers, which look like they're wrapping around the capillary. And so you can see there how it is that substances can get from the arteriole, so from the left-hand side of this diagram, through the fenestrations, in between, over the basement membrane, which is like a molecular filter, and then through those finger-like pedicels, and then in to the Bowman's capsule itself, and then we've formed glomerular filtrate. But I thought you'd needed to be able to sort of recognise that there. Okay, so moving on from there, let me just get rid of that. Okay, so I want to talk about the next process, which is selective reabsorption. And this happens in the PCT, or you'll have written there, proximal convoluted tubule. Convoluted just means, it's sort of coiled up, it's very long, and this is the mechanisms by which substances move are facilitated diffusion and active transport. So you need to know that. So, what's happened is that things have been forced out from the blood into the Bowman's capsule and into the proximal convoluted tubule now, just by size. So it's quite indiscriminate, really. And of course, that includes substances that are essential for us, like glucose, to name but one of them. And so we need to actually reabsorb, we need to selectively reabsorb certain substances like glucose, and so that's where it gets its name. And where are they selectively reabsorbed to? Well, they're reabsorbed into the blood supply, which surrounds the whole of the nephron. You might be able to see it sort of around the diagram, and because it's very richly supplied with blood here, I'm pointing to it here, and those blood capillaries are known as the vasa recta. So let's have a little look at what the cells are like in the proximal convoluted tubule, because we've got another type of epithelial level, so another layer of single cells, but this time they're cuboidal in shape, okay? And you can see, so they're not flattened, they're cuboidal. And you can see here that the cells themselves have some microvilli at the one side of them. So we've got microvilli here, generally providing a very large surface area. It's called a brush border. I'm sure you've heard that before. The cells are well supplied with mitochondria, so we know there's active transport going on there. There's ATP being generated in aerobic respiration. And you can't see it so well in this diagram, but you will see it in another. There are basal channels at this side as well, just to increase the surface area again. The cells are very, as I've said there in the information I've just put up, the cells are also quite tightly packed, and they're tightly packed so that substances can't seep between the cells. You know, it's just the substances that are moving through the brush border that will go into the cuboidal cells. Okay, so lots of features there, basal channels and microvilli to increase the surface area of the movement at the places you find those in the cell. And of course, then on the left-hand side, as we see, looking at that diagram there, the basal channels are really close to the blood capillary, and you know, obviously there'll be gaps there, pores there for substances to move on through. So a range of functions there. Okay, I've just popped in there which molecules will be reabsorbed for you to have a little look at that, because you know, I've sort of mainly mentioned glucose, but obviously there are other things as well, like salts and amino acids and water. So just to sort of summarize those adaptations of the PCT, the proximal convoluted tubule, very large surface area, cuboidal epithelial cells, numerous mitochondria, tight junctions between the cuboidal cells, and a very close association with capillaries. Well, why large surface area? Well, the length of the proximal convoluted tubule, the fact that there are millions of them in any kidney anyway, and further sort of instances of large surface area, because the cuboidal cells, as I've said, at the one side have microvilli forming the brush border, and the basal channels at the other side to increase the surface area for movement substances. You know, the mitochondria are there in order to provide ATP for active transport. The tight junctions I mentioned to prevent any movement, any diffusion, any backflow of substances. And the last one, obviously you need that close association with capillaries so that substances can actually effectively, efficiently be reabsorbed. So, okay, let's have a little look at those cuboidal cells then, because what we've got here, now I've put this diagram in deliberately because it's around the other way, and you've got to get used to seeing things from a sort of a, you know, a different perspective really. So what we've got here are the sort of amino acid and glucose here in the proximal convoluted tubule, and obviously we want to selectively reabsorb these, and so they are moving first of all by co-transport into this cuboidal epithelial cell, and then what happens is they can move out of that cell towards the vasorector, the blood, into the blood plasma, either by facilitated diffusion through a channel or secondary active transport using a sodium-potassium pump there. So there's a lot of information there. As I say, you know, you can look at that, you know, a little bit later when you have the time. But it's important that you can see there that there's co-transport going on. There's facilitated diffusion, which remember occurs via a protein channel, osmosis, because water will move by osmosis. So a whole host of processes that you need to be very clear of, okay? While I'm talking about proximal convoluted tubule, I'm just bringing that idea, even though I'm not going to talk a lot about this idea of type 1 and type 2 diabetes, because a classic sign that somebody has diabetes is the appearance of glucose in the urine. So the glucose is not being selectively reabsorbed, as I've shown you there. And that's because sheer number of those transport proteins in the membrane there. There is such a high concentration of glucose in the blood that it's being pushed into the proximal convoluted tubule, and the protein channels cannot cope. So you will see some glucose appearing in the urine, and I've just popped in there that idea of the differences between type 1 and type 2 diabetes for you to have a look at as well. So moving on, I know we're moving on at quite a pace here, but let's look now at the next part of the nephron, and that is the loop of Henle, or loop of Henle, depending on how you want to pronounce it. So we've got a variety of different processes going on here, but we know that this is called a hairpin. countercurrent multiplier. So the fluid is moving in opposite directions in the descending and then the ascending limb. So that's why it's called countercurrent. It's a multiplier because the solutes are becoming more concentrated towards the apex of the loop of Henle. It's called a hairpin because it's got a sort of a hairpin shape as well. So the medulla of the kidney, as I've said there, which is the inner part, remember it's the outer cortex and the inner medulla, that is becoming very concentrated with, you know, salt, different sorts of sort of sodium and chloride ions. So there is a very low water potential there. And the longer the loop of Henle, then the more concentrated and the lower this water potential can be. You can see some figures, you know, on the side as you track down that descending limb. Okay, and you will find in sort of desert animals that they have very, very long loops of Henle to concentrate water, as you know, we'll come on to in a little bit. So let's have a little look at the features there. Again, you may recognise this diagram here. I'm sure you've seen something like this where you may need to recognise the parts of it. And we've essentially got a nephron there with its collecting ducts on the right hand side, and we've got our descending limb, which you need to remember has is permeable to water, so water can leave it by osmosis, and it's possible for sodium and chloride ions to enter it by facilitated diffusion. Now contrast that with the ascending limb. It's not permeable to water, so water can't move in or out, and what happens is that sodium and chloride ions can actually leave by facilitated diffusion. They tend to leave by facilitated diffusion down the bottom and active transport as you ascend the ascending limb. Okay, so we find that the filtrate gets more and more concentrated as it goes down the descending limb because water is leaving it. And then as you go up the ascending limb, because it's impermeable to the water and the ions are transported out first by diffusion, as I've said, and then active transport, the fluid in there as you go up the ascending limb gets sort of less concentrated. Okay, now I wanted just sort of a couple of to show you a couple of what happens at different parts, but again I'll sort of leave that there for you to sort of check later as like a little summary. And I wanted now to sort of focus in on you know, a list of things that are happening in the loop of Henle before I move on. So what's happening here is that the job of these loops, and don't forget there'll be millions of those in the kidney, the job of them is to concentrate salts in the tissue fluid so that there's an osmotic flow, as I've said there, of water out of places like the distal convoluted tubules and the collecting ducts into that medulla there, okay, and from there potentially into the vasa recta and to be taken back to the rest of the body. So a few things here that you need to remember for any question that you're given really. We've got sodium and chloride ions actively pumped out of the ascending limb into the tissue, so there's a low water potential. Because that ascending limb is impermeable to water, while the descending limb is permeable, you will get that low water potential, you know, in those areas outside. And so water is going to leave that descending limb because it'll go from a high water potential in the descending limb to a lower water potential outside. So water is going to leave by osmosis and, as I've said, then get carried away in the vasa recta to the rest of the body. So the descending limb is becoming more and more concentrated as water is leaving it, as you reach the sort of the tip or the apex of the loop. And then as the filtrate passes up the ascending limb, it's going to become more and more dilute because it's going to lose ions but not water, remember. So I just thought it was worth just sort of summarising all of that on one sheet for you to have a little look at. And of course, you know, I'd mentioned there's this osmotic gradient where you'll find that desert animals have very, very long loops of Henle, so they can concentrate, you know, the water or sort of rather, so the water can actually be taken away and conserved by the body, taken away in the vasa recta. Okay, so that is quite a whistle-stop tour of those structures there. So let's have a little look now at osmoregulation. So osmoregulation occurs by something called negative feedback, which I mentioned at the start. And I've also sort of mentioned this briefly so far, which is that there are receptors which detect any change. In this case, there's a part of the brain you can see there, the hypothalamus, the master gland there, and it has receptors we call osmoreceptors. They will detect a low water potential. All right, so if there's a low water potential, a low psi there in the blood plasma, they will detect that and they will immediately then react to that. They will send a signal to the pituitary gland. Now, I've said there is the posterior lobe. You can get away with just saying pituitary gland, so don't think you have to say which particular lobe. And if you can't remember, don't have a guess, just write pituitary. Okay, so it's the signal then sent to the pituitary. That's the coordinator that will actually secrete the ADH. And the ADH is antidiuretic hormone. And what happens is that because it's a hormone, it will then be carried in the blood to the collecting duct walls and also to the distal convoluted tubule. And I'll say a little something about it binding with proteins shortly. But ADH prevents what we call diuresis. Diuresis is a condition where you produce large volumes of dilute urine. So ADH stops that. It prevents that happening so much. So I like to think of it in a way as sort of conserving water, preserving water in the body. So the ADH then is carried to the collecting duct walls, and these are the sort of effectors, if you like, okay? And it causes temporary aquaporins. So these are protein channels. We've got temporary and permanent protein channels there, but these aquaporins are sort of, they're present in vesicles, and what happens is the ADH causes the aquaporins to be added to the membranes there, and ultimately more water is reabsorbed by osmosis. So of course then what's going to happen if more water goes back into the plasma, the set point is going to be reached, and so the osmoreceptors in the hypothalamus are going to detect that, so less or even no ADH is released. So, you know, set point is released, is reached, and so ADH is not needed to sort of have its effect again, or as much a greater of an effect. So a little bit on the aquaporins. I think I've probably explained this already, but you can see there that there's aquaporins in red in a vesicle there, and antidiuretic hormone causes them to fuse with the membrane. So there's more channels available for water to move from the collecting duct into the blood. Okay, I just thought in terms of negative feedback, I'd say a little something here. So let's look, we've got a normal water balance then. To summarise, what happens is the water potential becomes more negative in the blood plasma, and so what happens is pituitary makes more ADH, and then what's going to happen is we're going to reabsorb more water because of the aquaporins, so we produce a small volume of concentrated urine. So we're just relating it now to the urine produced. And the converse of that is that if the water potential of the blood plasma becomes less negative, there's going to be less ADH made, and so the kidneys are going to reabsorb less water, so you're going to have larger volumes of more dilute urine made. And sort of that is the effect of ADH in terms of negative feedback. I've just popped this in as well so that you can see it's just there for me to say to you, don't forget that actually ATP is needed as part of this mechanism. So it's another use of ATP in the body. And I'll just finish this little bit of explanation part of the session by saying, yes, we produce, as mammals, we produce urea. It's less toxic than ammonia, as I said close to the start, but it does require ATP to eliminate it. Fish, because they're aquatic, they live in water, they can get away with producing ammonia. It'll actually diffuse out through their gills, and it'll then be carried away with the water as they swim away from it anyway. And birds and insects, they actually produce uric acid. That's non-toxic, and you don't need a lot of water to eliminate it, so that's ideal for survival in a dry environment, as part of terrestrial life. And of course, I can never resist popping in a little image of an orangutan, as they are mammals after all. So what we're going to do now is we're just going to have a little look at some typical questions as far as this topic is concerned. So, as I say, I will give you some short pauses, but the idea will be that if you're listening to this later, then actually you can pause, you've got time to think, and then you can reveal the answer for yourself. So the vessel that brings blood to the kidney is... it's the renal artery, and that branches into arterioles. And then two structural features of the glomeruli, that allow ultrafiltration to occur. So we talked, about a number of those, but if they needed two, then I'm just going to reveal those, and they are, I've put three there: gaps in the capillary endothelium. So the fenestrations, the basement membrane acting as a molecular sieve, the efferent arteriole having that smaller diameter lumen than the afferent, all of that, remember, creating that arteriole system there, creating the high pressure. And then this question at the bottom there, ultrafiltration in the glomerulus results in the production of glomerular filtrate, as I've said. The pie charts below show the percentage composition of solutes in human glomerular filtrate and in urine. We can see the pie charts there. There's the key. Using your knowledge of processes occurring in the nephron, explain the differences in glucose concentration between glomerular filtrate and urine. So even though they give you more, they want to know about glucose. So we can see glucose is present in the glomerular filtrate, but not the urine. So actually you need to supply enough information for 2 marks there. And I'll just pause while you think about that. And then the answer is all of the glucose is selectively reabsorbed. Obviously, we're not talking about a diabetic here, and that takes place in the proximal convoluted tubule, and actually it can be reabsorbed by active transport. Okay, so there's a number of things that you might be able to say there. Next question then, the urea concentration of urine is much higher than that of the glomerular filtrate. We could see that in the pie charts. Describe the role of the nephron and the collecting duct in achieving this increase in concentration. So we're concentrating the urea, okay, in urine. So I'll just pause again while you have a little think of the sorts of keywords that you might want to include in this answer, and there's 5 marks, so there's quite a few things to pop down there. So let's have a look. So quite a lot of information there. Less water being removed from the filtrate and reabsorbed. Urea, remember, is reabsorbed. It appears in the urine. Salts are reabsorbed in the proximal convoluted tubule, and of course that means water is reabsorbed into the blood plasma as well. There's active transport of sodium ions out of that ascending limb. Water moves out to the descending limb by osmosis, and that causes a low water potential in the medulla, and so draws water from the collecting duct, and then it's reabsorbed into the plasma. So all of that causes the urea that is still in the filtrate to be concentrated because there is less water there. And then suggests an advantage to mammals of excreting urine with a high concentration of urea. So let's have a little think about that. Okay, and here we go. I'm just going to reveal. So we've got less water being lost from the body in the urine. And, you know, think about it, if you're a terrestrial animal, then actually you need to guard against dehydration, and that's especially important, isn't it? You know, and you know, one of the mechanisms where we know we're getting dehydrated is we'll suddenly feel thirsty. So that's where you know you need to drink. Okay, next question. So I've show you a variety of questions here. You may be lucky enough to have a few marks for just labelling parts of the nephron here, and you can see we've got X and Y blood vessels. You've got our main parts of the nephron there, and the first thing it does, it asks you to name A, B and C on the diagram. So I'll just pause. Collecting duct A, proximal convoluted tubule B, C, distal convoluted tubule, and then it says pop some arrows in. Where would you expect ultrafiltration to occur? Did you think here, in which case that will be fine? Anywhere between that glomerulus pointing to the Bowman's capsule will be fine, and selective reabsorption point to a part of the proximal convoluted tubule. Okay, so the sort of, you know, other questions, name X and Y, renal artery and renal vein, and then explain the loop of Henle. Well, all I'm saying here at the moment is we've done that, so that is the point at which you can recap for yourself. And how, next question, how hormonal control affects the final concentration and volume of urine when somebody's dehydrated. Well, I've said something about that. There's a low water potential in the blood plasma. That's detected by osmoreceptors. You secrete ADH by the pituitary. The aquaporins are inserted into the collecting duct walls, and water moves by osmosis into the medulla and then the vasorecta. So what happens is your urine is small volume and concentrated. I wanted to get to this question because this is another type of question where you can sometimes be shown microscope slide images, and in this case they want you to actually identify the area of the kidney. And at the top diagram A, we can see these round structures here, which is the glomerulus and the Bowman's capsule, and we can't see them in B. So the answer I'm just about to reveal in A, so I should just pause. A is the cortex, the outer cortex, where you find the glomeruli, and obviously, you know, I've mentioned that is the reason there. Whereas B, you can just see that there are circular tubes. There's no evidence of any glomeruli there, so it has to be the inner medulla, you know, for the reasons I've said. Okay, so the nephron and how it's adapted for ultrafiltration. So again, I'm going to sort of pause there because I've mentioned all of these things before, so I'm just going to pause before I reveal. And you can see there what we talked about, the high hydrostatic pressure, the afferent arteriole with the wider lumen, the glomerulus with the fenestrations in the capillaries, the basement membrane as a molecular filter, and podocytes. And then this question below it, patients with severe kidney disease may be told to follow a protein control diet with low sodium chloride intake. Using knowledge of the role of the kidney suggests why the diet is recommended. A protein control diet will obviously, if again I'll pause, but actually what I wanted to say was that actually if you've got a high protein, you'll produce more urea and you can't remove that. So that is why you'd expect the diet to be protein controlled, and the low sodium. And the reason for that is then if you've got low sodium, then you're not having to remove excess salt. What I'm going to do now is I'm just going to go through the other questions, but as our time is not far from being up, what I'll just say is that all of these questions now, as you can see, I'll begin to sort of reveal the answers, but some of them are coming back again, and so you can test them for yourself. I will just say about this question here, this is a graph and it's very typical in that it'll say describe. So remember, describe is say what you see, and I've given you some examples of the answers there. And so I usually finish with one of these dice sheets. So what I'll do is I will leave you with this dice sheet where actually what the idea is that you've got your questions and then you've got a template to make your answers, and I can give you the answers to those next time. And the one last thing that I will also finish with, if you're used to my sessions, and that is picking 3 parts, as I mentioned at the start, and then picking out a range of keywords in the order that I ask the question, so that you can actually then pop yourself an answer together with all of these keywords in sentences in the sequential, in an order then. So I'm sorry that's been a bit of a whistle stop. I tried to do as much as I could. At least now you'll be able to see the answers if you go back through at your own pace later on. And just to let you know that the next A-level catch-up session is next Thursday, same time, same place. This one is on the nervous system. Thank you very much for listening. Best of luck.